In this video, we will discuss about how they come up to the point that Lectin can be used as a potential therapeutic target for pancreatic cancer. So first of all, we will understand the hypothesis that they have tested that usually comes up into their mind from the results that we have discussed in our previous videos. So what are the hypotheses that comes into their mind? The hypothesis in general form, or in simpler terms, the hypothesis is that the pancreatic cancer cells express some of the glycoproteins that are highly specific to the Lectin, and the glycoproteins that they are able to find out are that are highly specific to the Lectins are ITGA3 and ITGB1. They have the sialic acids, and this is more specific to the Lectin. Other commercially or other Lectin specific glycoproteins are these CD24s, and there could be other sialic acid associated glycoproteins that can interact with the Lectin. So, and these interactions will cause the suppressions of this phagocytosis that is caused by the myeloid cells against the tumor cells. So, in order to cap all the glycoproteins so that they cannot interact with the sialic acid, the easiest approach that they think about is that to cap the Lectins on the macrophages, so that all the glycoproteins that can interact with the Lectins, these interactions of all the glycoproteins with the Lectins can be suppressed. So this is their idea. However, the problem in this is that there is not so much high specific Lectin antibodies are available. So the researchers decided to make their own antibodies against this Lectin, and they make several antibodies, around 200 clones. Out of this, some of them they have selected for the ELISA, and we can see that the specificity against Lectins is significantly higher, whereas for the control they use Lec5, where the intensity is lower. And once they see the high specificities from the ELISA, they make the hybridoma cells of these antibodies, and then use the supernatants of these hybridoma cells against these CHOK1 cells that express Lectin, and also for a control they use CHOK1 cells that express Lec5. So what they did is they transfer the supernatant of these hybridoma cells of these Lectins antibodies into these CHOK1 cells with Lectin and Lec5, and then use the secondary antibodies with this 488 conjugated fluorophore, and then use the flow cytometer to measure the signals, and they are able to see that some of the clones gives high mean fluorescence intensities, indicating that they are more specific to the Lectin, whereas their control, that is Lec5, shows the low intensities. And out of this, they selected 10 to 15 clones for their further studies, or for their studies that they have mentioned in this panel A. So in this case, they use the they check the phagocytosis effect against this ASPC pancreatic adenocarcinoma cells. So they took the macrophages and capped the Lectins on the macrophages using the antibodies. And along with, for control, they use isotype controls and anti-CD24, which is also known to interact with the Lectin, and the commercially available anti-Lectins. However, the phagocytosis effect is significantly higher for the in-house developed antibodies against Lectins. And out of this, they have selected top 5, and then use them against the use this top 5 for the further experiments. So in the further experiment, what they did is they took the ASPC PDAC cells and the macrophages from different donors, and kept those macrophages with this in-house top 5 selected in-house developed anti-Lectin antibodies. And out of this, all the 5 shows a high phagocytosis. However, they selected the top one, that is 68A11A1, for their further studies. The results for all the 5s are also shown here. In this case also, they are able to see that the 68A11A1 shows a high phagocytosis, and they took these antibodies to do a more complicated studies, and before that they checked the ELISA, and they are able to see that it is highly specific only to the selectin proteins, and it is not specific to the other histec irrelevant proteins or CLF5 proteins. And once further identifying their specificities using ELISA, now they use the similar studies. However, they included the cancer-associated fibroblast also. So in that case also they are able to see increased phagocytosis for the 68A11A1. So here also they used all the top 5 antibodies that they have selected, and in this case also they are able to see that 68A11 shows a sorry about the confusion, this E, F, G figures are shown 2 times. So we have already discussed that from the ELISA they are able to see, so the 68A11A1's ELISA results are shown in the panel G, where we can see that the results are highly specific to the selectin proteins. So now they move to the more complex studies with this 68A11A1 antibodies. So in these complex studies they co-culture the ASPC1, MIA-PK1 and PANC1 along with the cancer-associated fibroblast, and use the selectin cap macrophages, and they are able to see high specificities compared to the anti-CD24 which is used as a control. And in the panel I also the time-based assay also shows the high specificities for for donor 2, and their average area under the curve also shows a statistically significantly higher for the anti-selectin antibodies compared to the isotype control. So this results validate their hypothesis that if the selectin interactions with their counter glycoproteins can be prevented, then it might lead to the increased phagocytosis effect of the macrophages against the tumors. And one more thing that comes into their mind, that it is many times reported in the literature that these selectins also cause the reduced proliferations of the T cells. They wanted to check whether by capping the selectins whether the proliferations again revived or not. So they used the anti-selectin antibodies on the macrophages and then checked on the CD8 plus T cells, and they are able to see that in the case of anti-CD3 and anti-CD28 the signals are low, but for the isotype control signals are high, and also when they use the anti-selectin antibodies the signals against increases, whereas when they don't use monocyte the signals are low, and non-stimulated the signals are high. So this is also a control. And then they were quantified, and this is just a representation. After that they quantified, where they find out that when they use the anti-selectin antibodies the proliferations against revived compared to the control case where they use anti-CD3 and CD28 which does not have the monocyte. So whereas when the monocyte is present without anti-selectin the proliferations goes down. So once they validate that it can revive the T cell proliferations, they wanted to check whether the T cells have the same functions of releasing lytic granules such as granzymes or not. So in this case, when they check the granzymes, they can see that in the anti-selectin antibodies the granzymes productions is high compared to the other conditions, and these things they have quantified on the right hand side of the panel K, where it is validated that the granzyme productions is increased or revived when the anti-selectin antibodies are used compared to the case where there is no anti-selectin antibodies. So this in all together made a point that the selectins not only increase the phagocytosis of the myeloid cells, but it will also helps in reviving T cell proliferations and its functions such as releasing lytic granules against the tumors. So from this two positive outcomes, they made a point that selectin can be used as a potential therapeutic target on the macrophages in the tumor microenvironment to increase its immune response against the tumor cells. And at the same time, it will also increase the immune response of the other immune cells such as T cells. So this is what they have presented in this figure. So if you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time. And sorry about this confusion that I presented this panel EFG 2 times.